Excellent. Great. Thanks, Tony. My name is Wilbert Dwyer. I'm co-lead of the Physical Oceanography Community of Practice. And welcome to the October meeting of this specific community. Just a really quick introduction about IARPC. IARPC is the Interagency Arctic Research Policy Committee, which brings together leaders from many agencies, departments, and offices across the U.S. federal government to enhance collaboration on research in the Arctic. IARPC Collaborations is the public branch of IARPC and aims to facilitate interagency communication, coordination, and collaboration to advance Arctic science. IARPC is currently preparing for its next Arctic research plan, 2027 to 2031, and is currently inviting input from the community through the federal registry. Next Wednesday, October 15th, there will be a listening session where you can learn more about IARPC in general and provide input into the next research plan. Note that the session is going to be postponed if the government is still in shutdown, so keep an eye out for that. So the Physical Oceanography Community of Practice aims to facilitate the exchange of information and encourage new collaborations in the physical oceanography of the Arctic Ocean across agencies, and primarily through the organization of monthly webinars. And we have some exciting news that we have a new federal co-lead on the team, Jackie Clement-Kinney, who is unfortunately furloughed at this point, so she won't be joining the call. In addition to that, we're also recruiting a new non-federal co-lead of the Physical Oceanography team. So please apply if you're interested in helping out. And Tony just put a link to the announcement of this opportunity in the chat. So today we'll be discussing the transport of sediments and geochemical tracers through the Arctic Ocean, and to discuss how they can help us better understand sources and pathways of waters that make up the Arctic Ocean. We have two very eminent speakers. We'll start with Laura Whitmore, who is a research assistant professor at the International Arctic Research Center of the University of Alaska Fairbanks. She has a PhD in marine chemistry from the University of Southern Mississippi. And she is involved in several projects, including NABO's Geo-tracers and Mosaic. And she will discuss how the distribution of elements can help us understand the waters of the changing Arctic Ocean. So the presentation is about 15 minutes. If we have some time for quick Q&A afterwards, we'll open the floor for that. But then we'll hand it over to our second speaker, who's going to be Rainer Ammann. So with that, I'll hand it over to Laura.
Okay, great. Thank you, Wilbert. Thank you for that introduction. Here we go. Okay, I'm just gonna get the meeting controls hidden here and hopefully get into full screen. I did just have my internet fully crash, so I'm a little bit worried I'm going to be talking into the void here and not know. But I've got my phone close by just in case that happens. But do carry on without me if that does crash again. So as Wilbert said, my name is Laura Whitmore. I'm a research assistant professor at the International Arctic Research Center at the University of Alaska Fairbanks, and I am based in Fairbanks. Okay. I do live here in Fairbanks. My family lives, works, and explores on the unceded lands of the Lower Tanana Dene. And to reiterate some of what Tony has already said, I have a lot of gratitude for Alaska's First People who have in the past and currently maintain really deep relationships with the land and waters of Alaska. I am a chemical oceanographer, and at the International Arctic Research Center, I run the Trace Element and Water Chemistry Lab. In that lab, we have a thermo element to high-resolution ICP-MS. This is the tool that we use to make measurements of ultra-low concentrations of metals in seawater. In my lab is also a Seal Quattro nutrient analyzer. And this instrument is run under the Nutrient Analytical Facility. And while I am a co-lead participant in that facility, the credit really goes to Meta Kaufman, as she's our analyst and she intakes all of the samples. So that's kind of the physical space that I work in. And as my title alluded to, I'm going to be talking about distributions of elements and how we can use that to understand things like circulation. And I'll caveat this with my talk is kind of at a very introductory level, which I think is a good place to kind of launch off on this discussion of tracers. So hopefully we can go beyond this. I'm excited to hear some of the questions that you guys have to facilitate the conversation afterwards. So I always like to start my talk with why I study the Arctic. I am from Alaska, so the Arctic has been very like proximal growing up. But in terms of being a scientist, one of the things I kind of take is changes drive ocean changes and global climate changes. And beyond just the climate of it all, there's geopolitics that go outside the Arctic, and there's societal impacts beyond the Arctic, but also at a core level to communities in the Arctic. So there's a lot of implications of change in the Arctic, and understanding the fundamental science in this space allows us to be kind of more decisive about those changes. So one of the questions I spend a lot of time thinking about is, how do or will fluxes of material change across differing spatiotemporal regimes? And I'm going to break this down a little bit. When I say material, most of the time in this talk, I'm going to be talking about dissolved trace elements. But material could be sediment particles, it could be biological particles, it can be dissolved trace elements or dissolved gases, it could be isotopes. And when I'm talking about spatiotemporal regimes, I'm thinking across the space of the Arctic, so Amerasian versus Eurasian, or even in that Amerasian sector, what's driving differences between the Canada Basin and the Makarov Basin? When I'm thinking temporally, what is the influence of an annual seasonal cycle versus annual or decadal trends based on, say, atmospheric oscillations? And going further than that, can we tease the kind of temporal differences apart in kind of the long-term climate scope? So this question is important because it flows into this next question, which was, how does the supply and distribution of material drive ecosystems? So if we change levers to the sources of material, how does that impact what's living in the Arctic? And I'm not going to get there in this talk at all, but that's one of the motivators for understanding these kind of base level distributions of materials. So in this intro, this is kind of where I'm going really down to the fundamental level here. It's not exhaustive. I'm going to be asking two questions. Where does material come from and where does it go? On the side of where does it come from, we have oceanic material entering the Arctic Ocean through the North Pacific and Bering Sea, through the Bering Strait there, North Atlantic Ocean waters, a whole bunch of freshwater discharge, 10% or more of global river discharge goes into the Arctic Ocean. We have seafloor sediments, especially on these broad, shallow shelves of the Bering, Chukchi, and East Siberian Arctic seas. And, beyond this, I'm highlighting these ones, but beyond this, there's also particles from the atmosphere and particles that have been entrained in sea ice, which might connect to, say, the atmosphere or seafloor sediments in some way. So where does it go? Once it's in the ocean, what are the pathways or the processes that impact the material? And the one that this group is all probably very familiar with is, of course, circulation. And I've got this little animation on the side that goes over a very few subset of the potential pathways that waters might take. So starting from the Pacific, water can come in, go through the Barrow Trough, get caught in the gyre, and maybe eventually come out through the transpolar drift and Fram Strait. Maybe it just circulates for a shorter amount of time, goes through the Canadian Arctic Archipelago on its way out. Surface waters can come in from the Yenisei and the Ob and rivers in that region, and maybe get entrained into the circulation of the Nansen and Amundsen Basin before going out again through the Fram Strait, maybe getting pulled again into the transpolar drift. You'll see the transpolar drift is a big theme here in these surface waters. Waters from the Lena often will get shuttled along the continental shelf before they get picked up into the transpolar drift as well, move offshore. Atlantic waters, these are deeper water once they're entrained in the basin, so they might get entrained into the basin and then go around the kind of rim current of the Arctic. So beyond circulation, material can get taken up into the food web. It can maybe regenerate back into the system or be exported to seafloor sediments. This is just another way to look at this. This image is modified from the Geo-tracers community. On the left-hand side of the image are these sources, so where material comes from, and I don't need to reiterate those. On the right side is where material goes, so either being kind of uptaken into biological processes, regenerating back into the water column, or getting exported through burial or circulation. Okay, and what drives changes in these sources? This is, again, not an exhaustive list. A big one is atmospheric conditions. So if you have a high pressure system sitting over the Arctic, you might get more export of materials out of the Arctic through the Fram Strait, be that sea ice or surface water. In reverse, if you have a low pressure system over the Arctic, you might get longer retention of those surface waters and sea ice in the central Arctic. So seasonal melt, sea ice or terrestrial, if there's, there's times where there's peak discharge, that might impact what's entering the Arctic. And then there's processes like Atlantification, which is not decoupled from either of these other two. Atlantification being the influx of anomalously warm Atlantic water circulating around the Arctic. There's lots of evidence that that warm Atlantic water is shoaling as well, which influences the kind of seasonal melt processes that are occurring in the Arctic. And then in terms of extreme weather patterns, I'm throwing this out there because we might get more storm events. We might get different effects of extreme weather patterns as sea ice retreats. And one of the big things is over these shallow shelves, if you have, say, a large storm system come through, you can resuspend a bunch of sediment, and that's a new source of both particulate material and dissolved material that was stored in those sediments. So all of these have the effect to change, again, not exhaustive. So how does all of this background material, in terms of looking at sources and processes that influence material, how does this manifest in our observations? And at this point, I'm going to go into some of the work that was done with Laramie Jensen as a lead co-first author on one of our papers that was published this year. And I'm starting with this section plot here. The next slide is going to have a lot of these, so I'm going to take a minute to just talk about the axes. The y-axis is depth in the water column. The x-axis is latitude, moving from the Beaufort Sea over to the Barents Sea. And if you look on the map, this is kind of like red shaded area. That's the transect we're looking at. The color bar here is silicate, and that's what's going to change on the next slide. There's going to be lots of different color bars for different parameters. But the point of this is that when we start to ask, what happens when you increase or decrease different supply of different source waters, how can you observe that in the Arctic Ocean? Well, our geochemical tracers are pretty acute for some of these things. And silicate's a great example because in Atlantic waters, it's very low, but in Pacific waters, so this upper halocline layer, it's very high. So you see this really clear signal of what might be Pacific origin or shelf-influenced waters in the section plots. So when we look at this for a big suite of elements, you can see silicate and zinc are quite similar. They have this high concentration in those Pacific origin waters. Barium also has a very high concentration, but barium's a little different from zinc and silica because it's also very high in the surface. And that's largely the influence of rivers. They tend to be very high in barium. So that influences barium in a way that it doesn't quite influence these kind of micronutrients and macronutrients. Iron is, I like showing this one because it looks so different from the others. Iron is highest in the Arctic in the transpolar drift. So that transpolar drift is picking up waters from rivers and the shelf, and it's moving very quickly across the Arctic Ocean. So it's picking up kind of like new waters and moving them across. And the iron signal in there is still very high. Likely because they're stabilized by maybe like ligands in a way that so it's not, you know, oxidizing and precipitating out as quickly. That's a possibility. So we can look at these, and in this 2025 paper, we looked at a massive suite of elements and said, okay, what patterns do they have against salinity, a very well-known conservative tracer? What patterns do they have, and what does that tell us about how they've been influenced by their sources or the processes that they encounter? And we saw more or less two main distributions, and then this third one is a combination of the two. So either they're really mixing dominated. It doesn't seem like as they pass over the shelf in this case being the Chukchi and Bering Sea shelves, looking at that Pacific water coming in, they're not getting change in their signal all that strongly. Theme two being there's a big change in their signal as they go from the shelf into the basin. So looking at some real data, these are some of my favorite tracers to show this example on. Dissolved gallium is an element I've spent a lot of time looking at. It is in the Arctic semi-conservative, and you can see that in this distribution because where the Pacific end member is. So the Pacific end member is quantified in this case as data that was collected in the Bering Strait. So waters coming in from the Bering Strait have this signal in this red box, and waters in the Pacific, the data behind that, waters in the upper halifine, the data behind that, match that signal. So there's not a change between what was in the Bering Strait and what is in the basin. The green box, that's the Atlantic end member. The shelf influence, you can see it quite differently. That red box that indicates the potential range of the end member is much lower than what we observe in the basin. So there's been some modification between the Bering Strait and below. Theme 3, barium, this is a combination. The reason we put this in combination is because there's a big effect of mixing, of physical mixing between these high concentrations of the river and the Pacific end member, but there's clearly also some addition of barium relative to what we expected the end member to be. Okay, you can do this for dozens of elements, dissolved particles, gases, isotopes, and we did that. And we were able to really, most of these fit fairly well into these three scopes. I would say most of them are shelf influenced in some way, which is interesting to know when you start applying them as tracers. So once you do this, once you look at these all qualitatively and say, okay, this is the pattern that this is the behavior that it has in the ocean, what do you do next? This is when I hope that you think, okay, I've done all this qualitative work, it's time to quantify. And what you quantify depends on your question. I've been largely giving the example of water masses in this, so I'm going to stick with that. But maybe you're not interested in quantifying water masses. Maybe you're interested in quantifying particle export or something more involved in the internal cycling of the elements. There are ways to do that too, but what I'm going to focus on in the next few slides is this question about water masses. So for the case of water masses in the Arctic, there are many water types, but we often simplify into 4 main water masses, being Pacific water and Atlantic water. I've shown in the map below in the red and the blue. And then sea ice melt formation and meteoric water, where meteoric water is really precipitation, but that's largely through terrestrial runoff into the Arctic. So on the side here, I've got my potential temperature and salinity plot, and I've got 2 water types, A and B, which you might consider to be Pacific and Atlantic water. And a linear mixing model is a way that we use our chemical tracer data to tease apart what water mass something is. So in this case, I know this is super simple, but in this case, if data fall on the line in between, they're behaving conservatively. And if we have a sample here in the middle, that's roughly 50% water mass A and 50% water mass B. And you can solve this. You have two unknowns, you're wondering how much is A and how much is B. That's this equation here. But to solve this, you need a second equation. So we use a tracer equation. In this case, I'm using salinity as my tracer. To deconvolve 4 different water types, you need 4 equations, which means you need 3 tracers. And we often will be using salinity, oxygen isotopes of water, and a nutrient-based tracer. And I'm not going to go into those tracers too in detail. That's something we can talk more about in the discussion. But what I want to highlight is that a linear n-member mixing model has a couple of assumptions baked into it. The first is that your tracers behave conservatively so that they will fall on a straight line between A and B. It also relies on fixed characteristics of the n-member. And this is something that I think is quite complicated in the Arctic because we don't always have the data we want to resolve the n-member. And there is potentially a range of the n-member through seasonal variation. So that's a really interesting thing to think about in terms of, is this an assumption that we can really make and be confident in? So that's where my call to the discussion is, how can we improve quantification of Arctic water masses? And I say here with increasing tracer observations, and what I mean by that is there is a lot of work to observe tracers right now. So with the tracer observations that we have, how can we improve our quantification of the water masses and water distribution? So I have a few suggestions. The first is we have 4 unknowns, we have 4 equations. What if we overdetermine the system by adding more tracers than necessary to deconvolve? We could evaluate new tracers. So a lot of geochemical tracers kind of fall into this category of semi-conservative, meaning we know there's processes that affect them, but the way that they're distributed in the Arctic is fairly inert. There's not a lot of change over their lifespan in the water column. But we could also evaluate other tracers, hoping to get somewhere closer to that conservative, and maybe that's through the measurement of isotopes of our elements rather than concentrations. We could consider ways to lean into the fact that our n-members might be changing, so they might be non-conservative. And the suggestion that I have there is maybe a linear inverse model becomes an option. And then finally, in like the most complex or the most degrees of freedom that we might have would be we could leverage complex regional models that are fully coupled with biogeochemistry. And there's a lot of work happening on that front in the Arctic. Even here at the International Arctic Research Center, Claudine Howery is working, and her group is working a lot on developing biogeochemical models for the Arctic. So I'd like to pivot with that call kind of being the end of my primary talk here, and just do a little shout out to the Nansen and Amundsen Basin Observational System. This program has been going on since roughly 2000, so almost 25 years now. And they are currently sitting outside of Nome, waiting for the sea state to be calm enough for them to take the small boat to shore at the end of their cruise. Nansen and Amundsen Basin Observational System has been doing mooring observations over the course of their observational period for salinity and temperature and physical parameters like currents. In the recent years, we've been increasing the hydrochemistry involved to really lean into that question of where are the water masses, and can we identify if this low salinity water is sea ice or river freshwater. So we've been really leaning into the hydrochemistry side. This year, what's really exciting is through the support of a lot of early career researchers, we also integrated some biogeochemistry and biology through DNA measurements, rate incubations of growth, O2 argon for net community production. They did vertical nets for zooplankton and microplastic. So I think all of this across this area shown on the map is going to be really interesting, because this is an area where you go from a Pacific dominated system to an Atlantic dominated system, and you can really see you're crossing the gradient, you're crossing not only Pacific and Atlantic, but you're crossing the transpolar drift. So there's lots of really cool things happening in this area. And the team's been out there for 55 days through the kind of complex situation of geopolitics. Their cruise was, their science time was reduced to 30 days, and they got a ton of the program done. So I'm very proud of them and taking the opportunity to give them kind of a virtual high five here. Okay, with that, I can pass it off to the next group.
Hey, fantastic. Thank you very much, Laura. This is very, very nice presentation. We're a little bit short on time, so what I propose is that we move over to Reiner and leave the discussion for the end. But if anybody has any questions for Laura, please put them in the chat to get the discussion started. So our second presenter is Reiner Amon. He's a professor at Texas A&M University, I think both at Galveston and College Station. He has a PhD in marine science from the University of Texas at Austin. And from 2021 to 2024, he was program director in the Office of Polar Programs at the U.S. National Science Foundation. So Reiner will discuss how CDOM can advance our understanding of halocline formation and freshwater sources in the Arctic Ocean. So with that, I'll hand it over to Reiner.
Yep, thank you, Wilbert. Good to see you all again. I'm sharing my screen and blow it up. I hope that works for everybody and everybody can hear me clearly. Scream if you can't. So as Wilbert said, I'm gonna give you an overview of basically 30 years of working in the Arctic since I moved over to Germany to do my, not my PhD, but my postdoc, and later on a research scientist. And so I'm gonna compile all that information, and it will focus on CDOM, which stands for color dissolved organic matter in the Arctic. And as you can imagine, this is only possible with a lot of support from different agencies and institutions which I have listed below this screen here. So what is CDOM? That is a picture of CDOM coming off the Siberian watersheds, and this tea-like substance that is discharged eventually into the Arctic Ocean. And that's what we try to use as a tracer for different things in the Arctic. How did I end up with this kind of approach? Initially, when I started my work in the Arctic, we were using ultrafiltration. That was difficult on a ship because we needed to have a 200-liter water sample, and most of my colleagues were not very happy when I consumed 200 liters from a single CTD. And so the frequency of sampling was limited, but here you see a number of samples we collected from different places in the Arctic. And so keep in mind, those are originally, they were 200 liters concentrated down to 75 mils. And so that's when you get that yellow substance back. Of course, in the 200 liters initially, you don't see anything, but this is what you get after the concentration. And you see 3 samples from the Greenland Sea, from the surface, from intermediate depth, and then from the deep. You see a slight decrease in the color. On the right-hand side, then you have a sample that was collected from the Yenisei River and concentrated, looks like Coca-Cola, more or less. And then we got one sample that we collected in the East Greenland Current. This is the current that carries all the Arctic surface waters towards the North Atlantic. So with your, naked eyes, if you want, you can already see what's going on there. The reason I show this is because, the Germans called the humic substances Gelbstoff, and Gelb is yellow, basically. And it triggered the memory of my colleagues at the RV, the physical oceanographers, when I showed them those samples on the ship. One of them remembered, hey, somebody built a fluorescence probe to track the Amazon River. But the person left, but I think the probe is still rolling around somewhere on the shelf back at the RV. And so we decided that the following year, we're going to bring this fluorescence probe to see if it helps us track this signal while the CTD is going into the water. And this is basically what we did. You're looking at data from the 90s here, collected during Polarstern cruises. All of it in the Greenland Sea along the East Greenland Current. And the data that you're looking at are oceanographic sections of fluorescence that is unique in tracking color dissolved organic matter. And you see very clearly at all the different sections at 79, at 75 degrees north, at 71 degrees north, and even down in Denmark Strait, you see very clearly this maximum of fluorescence coming in the center of the East Greenland Current. And that showed us two different things, right? First of all, that the Siberian River signal can be traced all the way to the Denmark Strait, which gives you a time scale of about 10 years, depending on what kind of transit times you're believing in. So it showed us that this tracer is at least stable over that time scale. And the other thing that we learned from that data set, that sedum signal is very strongly related to lignin phenols, which is a very unique biomarker to track terrestrial organic matter. So initially in the 90s, we thought, okay, now we have an in situ tool to track river water in the Arctic Ocean. Later on, as you will see, it turned out a little differently. Hydrographically, this is what the sedum signal looks like related to temperature and salinity. And if you believe that that sedum signal is mostly coming from rivers, you might expect that you see the highest values at low salinities, but that's not what we found. The maximum concentrations of sedum were always associated with intermediate salinities of about 33 and temperatures below -1.5. And so that are hydrographic characteristics that you would typically find in the halocline of the Arctic Ocean. 10 years later then, in the meantime, I've had moved to the US, and so it took me a while to set everything back up. But we did one study together with my colleagues that looked at satellite data to track the terrestrial organic matter in the surface waters of the Arctic. And that's what you see in that picture here. You're looking at a climatology between 2002 and 2009. And you see very clearly that elevated signal on the coast of Siberia, the Kara Sea, the Laptev Sea, and the East Siberian Sea, as well as on the North American side with the NSA, with the Mackenzie coming out. Of course, in the Arctic, you are limited in the use of satellites because of the sea ice. And so it took another 10 years to be able to compile actual observational data in the Central Arctic. And so in this case, you're looking at multi-national, multi-annual and multi-cruise data. You should be able to see all those black dots. So those are all actual stations that were occupied at some point between about 2005 and 2020. And on the left-hand side, you see the distribution of dissolved organic carbon, with that very large input from the rivers, as you see on the Eurasian side, and somewhat smaller inputs from the Mackenzie on the other side. But I want to focus on the right-hand picture here, which gives you the distribution of a proxy, an optical proxy, that we know does a pretty good job tracking terrestrial organic matter. And so what you see here clearly is what was pointed out before, that a lot of that Eurasian river signal ends up in the transpolar drift. after it stretches pretty far into the East Siberian Sea and then comes across the Arctic Ocean and eventually leaves mostly through Fram Strait, to some degree through the archipelago, but that hasn't really been quantified. So that is the overall surface picture that we saw from those optical tracers. And then what you see here is the vertical distribution of the fluorescence signal. And you see 3 different years. So on top you have 2005, where we crossed the Arctic Ocean on the Swedish icebreaker Oden. 2007 was a cruise on the Polarstern. And then 2015 again was a combined cruise of the Geo-tracers group, showing both Polarstern data as well as Heli data on the other side. And what you see, first of all, is that that Sediment signal is always there in the Arctic, and if you focus on the transpolar drift here, which is that large, deep red signal that you see, that is for you a very helpful tool to have if you're on the ship, because you immediately know if you are inside or outside the transpolar drift based on the fluorescence distribution here. The other thing that I want to point out in those figures here is that the Sediment signal is very closely related to the isopycnals. And so that shows you that there is a physical process that is very important in distribution of this Sediment signal, no matter where it comes from. And this, of course, would be exactly the density where you find the halocline. And so now we know that that Sediment signal actually tracks the transpolar drift for one, but it also tracks the halocline very well. And when I say this, I mean both the upper halocline from the Pacific side as well as the lower halocline from the Atlantic side. Where it becomes a little more complicated is when you compare the different metrics that we have. And so here you're looking at the 2005 data set from the Oden. And again, you're looking at the same distribution of Sediment on the bottom in 2005 and 2015, actually. But on top, you only see the data for 2005. On the left-hand side, you see the surface concentrations of Sediment. In the center, you see the surface concentration of lignin biomarkers. Again, remember, lignin is a very good tracer for terrestrial organic matter. And then on the right-hand side, you see the distribution of meteoric water, which is based on the water mass decomposition model that Laura has introduced in her presentation. And so if you look first at the transpolar drift, so the center of the Arctic, you find very high Sediment levels. You also find very high lignin concentrations, and you find a high value for meteoric water. So that makes perfect sense, right? You have a lot of river water that should have a lot of Sediment, and it should have a lot of lignin. However, if you focus on the Canada basin here, you see that there's very little Sediment in the surface waters of the Canada basin, basically everywhere. And so you have very low Sediment. You also have very low lignin phenol concentration. So there's very little terrestrial organic matter in there. But you still have a very high value for meteoric waters. And so something is off here. And so we started to think about this, and I will have something to say a little bit later on. But there's something that does not jive if you compare the different metrics. So what is the source of Sediment in the Arctic Ocean? So in the plot on the upper left-hand side, you see the relationship of Sediment fluorescence to lignin phenols, right? The same thing that I introduced initially from the East Greenland current. And in this case, we split the data between the Eurasian basin and the Canada basin. And so if you look at that plot, it makes perfect sense for the Eurasian basins. You have high lignin whenever you have high sedum or vice versa. However, in the Canada basin, that relationship does not exist. There's basically no significant relationship of sedum fluorescence to the lignin concentrations. The other thing that makes the whole thing even more interesting is if you look at the lower right-hand plot here, where we relate sedum fluorescence to the fraction of meteoric water or river water. And again, the data are split between the Eurasian basin and the Canada basin. And you see that in the Eurasian basin, it makes sense. You have higher sedum fluorescence whenever you have more river water. But in the surface Canada basin, you actually see the opposite. You have lower sedum fluorescence with higher values of meteoric water. Okay? So something does not quite match up. On the other hand, if you look at the lower left-hand plot here, where we relate sedum fluorescence to the fraction of sea ice melt, that seems to be the one metric that explains the pan-Arctic distribution of sedum the best. And that has to do with the fact that apparently sea ice formation and melt is the #1 determinant for the distribution of that sedum signal across the Arctic Ocean. The other thing that was interesting, that we also saw a very strong relationship in the Canada basin between the sedum fluorescence and the DIC concentrations. So with higher DIC in the surface waters of the Canada basin, you also get higher sedum fluorescence. And then we expanded that to compare sedum signals to the apparent oxygen utilization in the surface Canada basin. And again, you see a very strong relationship with higher sedum values at higher values for apparent oxygen utilization. And taking those two together now, we are pretty sure that that sedum signal in the Canada basin is derived from some sort of a decomposition process, most likely happening on the Chukchi shelf in the sediments, and then that signal gets carried out into the open Canada basin. And if I have time, I will show you some of the distribution in the entire Canada basin with some of the more recent data we have gotten. And so the main question that we had, what is the source of sedum? I would say it's pretty clear that in the Eurasian basin, the main source of sedum are the rivers coming in from Siberia. But in the Canada basin, the main source of the sedum is more likely to be a decomposition of marine organic matter that was produced on the shelf and gets decomposed in the sediment surface layers. All right, so how does this apparent oxygen utilization signal look like in hydrographic terms? Here you again look at different sections in the year 2005, 2015. In this case, we have two sections from 2015. And you see a couple of things. First of all, it's a very clear signal on the Canadian side, but it's completely absent on the Eurasian side. So it does not exist there. The other thing that was interesting is, in this case, obviously, it's only tracking the upper halocline, or Pacific winter water, if you want, and not the lower halocline. But it also seems that the signal is the strongest in the northern Canada basin, and it gets weaker when you go into the southern Canada basin. And so this might be a good way of tracking the halocline on the Canadian side directly from the CTD, because all you have to do is to look at oxygen saturation, which gives you the same kind of information as apparent oxygen utilization. And that's very, very helpful for the people that are collecting samples at those cruises. In terms of the CDOM signal in vertical, you can see in the left-hand plot that the top of the CDOM signal is apparently derived from the Pacific, and the bottom of the CDOM is apparently derived from the Atlantic, as indicated by this NO over PO ratio that was introduced by Wilson and Wallace a long time ago, where your bluish colors would be indicating a Pacific origin and your reddish colors would be indicating an Atlantic origin. If you look at the Eurasian basin on the other side, on the other hand, it's mostly determined by Atlantic water, with some Pacific water in there that most likely comes from eddies that made their way into the Eurasian side. This is just another way of showing those CDOM sections. Now we are not relating them to isopycnals, but we are relating them to isolines of the fraction of sea ice melt. And in this case, you might not be able to see it, but they are all negative, which means that you have brine injected into those layers, and that would mean that CDOM basically is part of the brine that was introduced on the shelves and that made it out into the open Arctic Ocean. And that brings us to the second question that we find very challenging is, what is the source of the meteoric water in the Canada Basin surface waters, right? Why does it look like river water but does not have any of the organic matter in it? And so we started out looking at the hydrography, and interestingly enough, even in the Central Arctic Ocean, you see that the CDOM is the highest at intermediate salinities, somewhere around 33, and a density of approximately 27 kilograms per cubic meter. And that you see, for the most part, a very clear decline of CDOM as you go into lower densities or lower salinities. And so that again would indicate that there's some sort of process that takes river water and fractionates the river water from the ice. That means that the water molecule and your organic molecules are separated by the freezing process. And that's why you see this very unique distribution where the decline in CDOM is basically caused by a dilution with sea ice melt or ice melt.
Right.
Another thing that was clear that if you look at the shelf waters, that you get a very wide variety of shelf waters coming into the Central Arctic Ocean with a density that can range from 24 all the way to 28. And some other indication comes from some work that our colleagues at the ARBI have done that relate river discharge to the volume of ice melt that you find in that system. And so in this case, they're focusing on landfast ice, and they show that you have, in terms of volume, as much landfast ice as you have river discharge during the freshet. And so that alone, am I pushing the time?
Yes, you are. We got 4 minutes left on this call. So,
all right, so let me jump ahead here to answer that question. So why do we think that the meteoric water in the Canada Basin is not river water? If you look at the relationship between Delo-18 and salinity in the Canada Basin and the Eurasian Basin, you see that the relationship looks very different, right? So your measured end member in Arctic rivers is -21 per mil for oxygen isotopes. But if you use the relationship that you see in the open Arctic surface waters to calculate the theoretical end member, then you get very different values. For the Canada Basin, you get a zero salinity end member for Delo 18 around -14, which is a lot higher than you would expect from the measured end members. While in the Eurasian Basin, you have the opposite. You get a lot more depleted values. And we think that that has to do with the fact that in the wintertime, on the shelf, you have a lot of river water. And so the ice that you form on the shelves contains a lot of river water, right? And that ice is then transported along the coast towards the Canada Basin and is melting sooner or later. And it looks like river water, but in the end, it really is sea ice melt that came from the shelves. And that would explain why it has a meteoric fingerprint in terms of oxygen isotopes, but no terrestrial fingerprint in terms of lignin and DOC. And so what we did, we changed the water mass decomposition model a little bit, the classical one, and replaced the nutrient tracer with a sediment tracer. And if you do that, you get very different results in terms of sea ice melt. So the classical results are shown below here, where you have very low values of sea ice melt. And the top panel here shows you the amount of sea ice melt if you use the CDOM as a third tracer. And you see that there would be a lot more sea ice melt with the modified model than what the classical model would show you. And just to, I'm gonna skip the new data and just get to the end here. So our outlook after 30 years is that if we get a higher resolution data set for lignin biomarkers, then we are pretty confident that we'll be able to come up with a better model to describe water masses and freshwater sources in the Arctic Ocean. The other thing that became clear to us that in order to really make progress to understand halocline formation in the Arctic, we need to make better use of the new underwater navigation technology that ONR has now established in the Arctic, and use that platform to get wintertime data to learn more about the halocline formation and the hotspots. And I stop right here.
Fantastic. Thank you very much, Rainer. This was an incredibly interesting presentation. I hope that your slides will be posted online as well. Would be very useful for us all to go back to and yeah, get some more insights in these processes that you have been describing and that are very well aligned with the stuff that Laura has shown us as well. We are at the top of the hour. Might be a couple minutes left for questions, but if anybody has any very urgent questions that they want to ask, then please raise your hand and we'll call in. Don't see any hands. If I may put in a plug, so as part of the Consortium for the Advancement of Marine Arctic Science, we're doing a couple of experiments with passive tracers, and Carlin, who is on the call here as well, she's leading one of those activities. Thea Weiss-Gibbons from the University of Alberta, I believe, she will also be coordinating some of these experiments. Carlin, do you want to talk about this real quick?
Yeah, absolutely. First of all, thank you for the really interesting presentation, Rainer. So yeah, as Wilbert mentioned, using E3SM Arctic and with our collaborators at CMOS, we're carrying out a series of passive dye tracer experiments looking at oceanic, meteoric water, sea ice tracers, as well as riverine tracers, and trying to do that in a way where we do kind of short-term releases so we can get an idea of the time scales of these water masses and their pathways and how they're exchanged throughout the Arctic. So yeah, really, really interesting to look at kind of on the observational side, some of these pathways. And it would be cool to kind of talk about comparing some of the model experiments with the observations. So, but yeah, that's what we're currently working on, kind of under the CMOS collaborative umbrella.
Yeah, and just to chip in there real quick, so one of the things that Colin and Thea are particularly interested in is the kind of the time dependence of the circulation and how that affects the distribution of the tracers. And another thing is that we're using kind of passive tracers, right? So these are volume tracers, but also salinity tracers. But yeah, looking at Laura in particular, the wide range of tracers that she's actually studying would be really interesting to see how the different tracers kind of behave differently, right, in terms of their decay or how they are being processed in the Arctic system. And yeah, as Laura also alluded to, some of these marine biogeochemistry models can help kind of model some of these behaviors, but I'm not sure if things like barium or some of the other more exotic traces are actually implemented in those models, and whether that would be a possibility. Are you aware of any of those models that do that?
Yeah, it's something that I've had in kind of short bursts of conversations with people, but I'm not aware of it kind of being applied directly in that sense. And something, one of the questions that I have, seeing, especially with Reiner's and my own data too, like I've swapped out that nutrient tracer where Reiner swapped it out for CDOM, I've swapped it out for gallium, where CDOM seems like it can really help resolve the kind of upper 500 meters of the water column. Gallium should be really good at resolving the Pacific source water from the Atlantic source water. And the question that I have lingering is, okay, so now I have two models that show me two different distributions of waters. Which one of these is more correct, right? And, seeing some of Reiner's plots showing, this kind of mystery in the surface water. Why isn't there high CDOM if there's high meteoric water? That's a fascinating question. And we can combine it with other tracers too, because that same section that Reiner showed from 2015, I have barium data from, and barium is high in that section, where that sea ice melt is high, but barium is low in sea ice, relatively speaking. So there's some mystery here. And I think it's a really, really interesting question. So not only when you replace a tracer, maybe you're getting better resolution in one direction, but not another direction. So how do we verify that fits kind of the real world distribution and not some operationally defined distribution that we've come up with? I know that went far from your question, but it's been rattling in my brain since Reiner started talking.
Do you have any response to that, Reiner?
Yeah, well, you need to understand the behavior of barium during sea ice formation, for one, right?
Yeah. I mean, the expectation that we have for a lot of the parameters, and I think sea ice formation and melt is where the linear model almost breaks down, right? Because the end member during melt should be very different than the end member during formation.
Yeah.
And the end member during formation is dependent on a lot of things, like the temperature of ice formation, the rate of ice formation. So in a linear scope where you've defined one end member, you really, that part almost breaks down where you can use that same parameter for formation and melt.
Yeah, we're hoping that using more equations maybe helps to,
Right.
kind of knock that out.
Yeah.
That's why
All
we're
right,
doing the
fantastic.
lignin thing. And, in the neighbors, the guys collected samples for lignin for us again. So we will have,
Yeah.
a pretty nice data set combining 24 in the Central Arctic with the 25 neighbors data on the closer to the shelf. So that'll be really helpful.
Yeah.
And hopefully another heli cruise next year.
Yeah, hopefully. I did have one comment that I threw in the chat based on your AOU slide, and it's a little nitpicky comment, but just because it might be interesting. There's a point in your plot that has really high AOU. And in our conversations through the Geo-tracers workups and synthesis meetings, it appears that there is an eddy there that year. So there was almost going to be a whole synthesis paper about the effects of an eddy kind of coming off the shelf and what that
The silicate is also higher.
Yeah.
So basically it means the eddy picked up that
Right. And
shelf
pH
signal.
and DIC is really
high.
lit up in that as well.
Yeah, there's lots of eddies.
Yeah.
There was not a single year that I was out there when I didn't see at least one eddy. On a straight shot. So they are very, very important.
All right, with that, and unless anybody has any other urgent questions, I think we should wrap this up. Very interesting presentations. Thank you very much, Reiner. Thank you very much, Laura, and also for the discussion. And well, this is certainly a lot of food for thought and for future research. So we're really excited about that.
Yeah, thank you for the invite.
Thank you.
Tony, anything else that we need to discuss, or are we good to go?
I think we're good to go. You covered some good updates at the top of the call. So thanks all for joining, for hanging on a little longer. Laura and Reiner for your presentations, and Wilbert for hosting.
Yeah, thanks for having us. And I'll see you, Guy, on some other meetings, I guess.
See you soon.
Yes, bye everybody.
Thank you very much. Bye-bye, everybody.